Hello, welcome to Group 51's redesigned SNP-CHIP test for Parkinson's disease. Group 51 chose to redesign the test for Parkinson's disease since it hits close to home for me. My father was diagnosed with early onset Parkinson's in 2015, and my family has been adjusting with that for around a decade. For those of you who do not know, Parkinson's disease is a progressive disorder of the nervous system. It begins by causing trembling in the limbs, including the arms, legs, feet, and face. The limbs and body can become rigid and stiff, and an infected individual may have impaired balance and coordination. Emotions and cognition are also affected and can lead to the development of psychiatric conditions. In the United States, Parkinson's disease occurs in 13 per 100,000 people. Most cases of Parkinson's disease occur in people with no apparent family history of the disorder. The cause of these sporadic cases remains unclear. However, of the approximately 15% of people with Parkinson's disease that have a family history, it can be traced back to mutations in the LRRK2, PARK7, PINK1, PRKN, or SNCA genes.
The 23andMe test checks for genetic variations through saliva samples to have a deep understanding of their ancestry, family health conditions, and traits. It is heavily focused on single nucleotide polymorphisms, otherwise known as SMP. One of the most essential variations tested by 23andMe is the BRCA1/BRCA2 gene mutation associated with the likelihood of breast and ovarian cancer. These variations can affect susceptibility to diseases, so proteins involved in repairing damaged DNA are encoded to maintain genomic stability. These same proteins are also produced in specific cell types, including breast and ovarian tissues. However, 23andMe does have its drawbacks, with one of the most prominent being lack of genes associated with traits or diseases. More specifically, it does not test for all known alleles of the genes examined. Although it provides valuable information, it does not go in depth into the associated genetic condition, so having a proper diagnosis from medical professionals is still the better option. 23andMe tests for two variants of the Parkinson's disease: the G2019S variant in the LRRK2 gene, and the N370S variant in the GBA gene. Here I want to do some research into the LRRK2 gene. The LRRK2 gene can impact both the structure and expression of the LRRK2 protein. The LRRK2 protein is involved in various cellular processes, including regulating the function of the other proteins and pathways involved in cellular survival and death. Changes in the LRRK2 protein can disrupt the cellular processes. It can lead to abnormal levels of the protein in cells.
Our test will be designed for those who have a genetic background with Parkinson's disease, i.e., if a parent or grandparent has been diagnosed with a genetic trait, like my family. Unfortunately, that only accounts for about 15% of people with Parkinson's, since so much is still unknown about this disease. For our test, we would look at more than just the G2019S variant of the LRRK2 gene and the N370S variant in the GBA gene. So many more genes can cause Parkinson's when affected, so our test will investigate more of those genes, including PARK7, PINK1, PRKN, and SNCA genes. Specifically, our test would hone in on the SNCA gene, which encodes alpha-synuclein, a protein that plays a central role in the pathology of Parkinson's disease. Alpha-synuclein is a major component of Lewy bodies, abnormal protein aggregates found in the brain of individuals with Parkinson's.
In addition to the LRRK2 gene, our test will also study the PARK7, PINK1, PRKN, and SNCA genes. Because of this addition to the test, we get to specifically study a protein coded from the SNCA gene. This is the main difference between our test and the 23andMe test. Alpha-synuclein compiles into clumps known as Lewy bodies. These Lewy bodies have been studied and are believed to be passed from one neuron to another in the brain, causing the spread of Parkinson's disease throughout the nervous system. With the SNCA mutation, 33% are shown to have onset before the age of 40, and an important characteristic is cognitive decline. The variant V15A of the SNCA gene was the first to be causally linked to a rare autosomal dominantly inherited forms of Parkinson's disease. This test provides more insight that 23andMe cannot provide. Although an abundance of research has been done on Parkinson's disease, we unfortunately do not know all the causes and science behind it. Since there are so many genes and factors that play a part, narrowing it down to one genetic testing factor may not be ideal for any individual, and therefore this test may not give you a definite answer on your susceptibility to developing Parkinson's disease.
An emotional concern of testing positive for a genetic disease can be family treating you differently. They can reject and isolate you for the rest of your life, so carrying that guilt about your results is a tough burden to have. Additionally, they could be less willing to support you financially throughout the process. Testing positive comes with multiple doctor visits, surgeries, and medications for several years, which can increase family medical expenses immensely. Some people don't believe that family attention is worth finding out the likelihood of them attaining a genetic disease. Some people believe that it may allow healthy individuals to be worried about the possibility of genetic testing, allowing them to be discriminated against for no reason.